Hi everyone, my name is Kiana Lee and I'm a fifth year plant science student at the University of Guelph. This summer, my research question was, what are the effects of reduced pollination on floral longevity? Global pollinator populations are declining due to impacts of climate change and habitat loss. In North America, wild pollinators have had a 40% decrease in population. This is significant because 80% of wild plant species rely on insect pollinators to set seeds and produce fruit. These declines can have severe impacts on plant biodiversity and agricultural crops that require pollination. Depending on the species, plant responses to pollinator declines can include increased nectar rewards, increased self-fertilization, and showier floral displays. One response to pollinator declines may be an adjustment in floral longevity. Floral longevity is the amount of time a flower is open and receptive to pollination. For example, on the timeline figure, the floral longevity is 10 days. Although this trait can be fixed or variable, changes in floral longevity can be costly in terms of time and energy. Investment in floral structures like petals involve large energy allocations for maintenance. Therefore, understanding floral longevity is critical in determining the implications of reduced pollination. I hypothesize that floral longevity will increase in response to reduced pollination. In the line graph, the x-axis represents total floral longevity, and the y-axis represents the amount of pollination. I predict that if decreased pollinator visitation decreases the pollen load received by plants, then increased longevity will maximize the length of time flowers are open and exposed to pollinators. I used an experimental method to evaluate my hypothesis. My study system, Lobelia siphilitica, is a perennial wildflower that is primarily pollinated by bumblebees. There is complete separation between the male and female phases of sexual development. To dispense pollen, flowers must be visited by a pollinator in the male phase before becoming female. For my field study, I set up experimental plots in the University of Guelph Arboretum. Half of the plants were exposed to normal pollination. I reduced pollination in the other half of the plants by covering them with mesh bags for half of the time that they were exposed to pollinators. I measured floral longevity by following 3 buds per plant through the entire floral lifespan. I measured total longevity, which is the number of days a flower is open in both the male and female phases. I also determined male phase and female phase longevity. In the following figures summarizing my results, the x-axis represents the normal and reduced pollination treatments, and the y-axis represents the measure of floral longevity. Contrary to the hypothesis, I found that there was no significant difference in total longevity between the normal and reduced pollination treatments. The median of the normal treatment was 8.1, and the median of the reduced treatment was 7.9. However, I did find differences in the floral longevity of the 2 sexual phases. Compared to the normal pollination treatment, male phase longevity was 13% longer when pollination was reduced. There was no significant difference in female phase longevity between the normal and reduced pollination treatments. I also noticed large variation in female longevity among the pollination treatments. In conclusion, reduced pollination did not increase total floral longevity. However, reduced pollination increased male phase longevity. If male phase longevity is increased, then pollen may be taking longer to dispense from the flower. Longer periods of pollen dispensing may limit reproduction because pollen may be released more slowly. It's uncertain why there was no response in female phase or total longevity. If male phase longevity is increasing, female phase longevity should decrease if there's no change in total longevity. My current analysis only uses some of the longevity data I collected from my experiment. In the future, I'll use the rest of my data to see how floral longevity responds to reduced pollination. Thank you for listening to my presentation.